Good afternoon and welcome to this edition of the Westwood Lecture Series. And we invite great colleagues across the university, across different disciplines, to come give us a lecture and do some Q&A with fellow colleagues here. This is a great part of the intellectual feature of this community of scholars. So today we have a very exciting and unique talk, and a personally very relevant one, given how much coffee I consume. And I know nothing about this leaf rust fungus. And it turns out, I just learned from our great dean here, Bernie told me we've got the largest collection of such fungus here at Purdue. And about 13 years ago, Professor Cathie Aime joined us in the Great College of Agriculture to study this and many other related areas. In fact, we just had the day-long first annual symposium of digital agriculture, and weekend hackathon organized by the students in Ag, and that was a great event as well. So we have a lot of wonderful things going on as always. College of Ag here. So thank you, Bernie, for your leadership, all the colleagues here, and a warm welcome to Cathie to today's Westwood Lecture. Thank you very much.
Thank you, Mr. President, Dean, all of my colleagues here today. It's, of course, a tremendous honor to be here to speak to you about one of the projects we do in my lab. I'm a traditionally trained mycologist, which means I study fungi. I study all aspects of fungi and many, many fungi. But one of my expertises lies in the study of rust fungi, which I'll explain a little bit about, and hopefully why I got interested in this particular project today, looking at the fungus that causes a really economically devastating disease in coffee. And before I start, I just want to make a couple of acknowledgments. Of course, everything I present today was not done by me alone. There is a huge team of people involved in this. First off is a woman named Liz Johnson. Liz is the head of what we call the Inter-American Institute for Cooperation in Agriculture. And she noticed this new disease in 2013 in Jamaica that was devastating their coffee farmers. And Liz gave me a call to try to help her get this under control. And that was really my entree into the world of coffee rust. I've had 3 really, really talented postdocs work on parts of this project. Amnat, Luis, and Terry Torres Cruz, who is still a current postdoc, leading our effort. And then other lab members, students and postdocs, Hector Urbina, Paula Gomez, and Jorge Diaz also worked on small parts, including getting our greenhouse studies up and running and our genome annotated. And then I've had numerous interns from around the world come to study coffee rust fungus in the lab and contribute to part of this study that I'll show you today. In addition to all these members, we do have a lot of collaborators on parts of the project that I'm not going to talk about today, but these people are also important in the overall picture of what we're doing with the coffee rust fungus. And of course, funding is always problematic. Initially, we got seed funding from ICA, from Liz Johnson. We've received funding from World Coffee Research, and then more recently from the USDA and NIFA to work on this problem. So, coffee. We all cannot live without it, or at least most of us. It's one of the main agricultural products on the planet. There are some accounts that say it is the #1 most traded agricultural product. It's certainly up there in the top 2 or 3 traded products in the world. In 2024 alone, the world consumed 3 billion cups of coffee a day. That's a lot of coffee. Just in the last 5 years, the wealth or the worth of this coffee has more than doubled, whereas in 2024, the coffee crop was estimated at about $460 billion. Demand for coffee continues to grow, especially in parts of Asia, where the market is booming these days. But production is on a downslide, and we have had production deficits in recent years, in the last 2 years. And there are doomsdays who predict that we will have massive shortages by 2030, and that as soon as 2050, coffee, or at least Arabica coffee, will be a luxury item available to only some people who can afford it. Important to keep in mind when we talk about coffee is that it's one of these crops that is mostly produced by small shareholder farmers. It doesn't, at least Arabica, when we talk about Arabica coffee, it doesn't lend itself to mechanization. The beans, or the cherries we call them, ripen at different times. People have to constantly move through the fields and pick them. They grow at higher elevations, best, and on slopes. So it's just not easy to mechanize. So we are looking at production by small shareholder farmers for most of our Arabicas, at least. 90% of this global production is in developing countries, which again can be problematic. 60 to 80%, again, as I said, is by small shareholders, and about 25 million of these small shareholders depend on this crop for their subsistence. These are mostly people who might come from a subsistence cropping background, but booms in coffee and organization by coffee boards in different countries, farmers are encouraged to turn all their acreage over to coffee production. And this leaves them very vulnerable, of course, to any failure in the crop. Now, when I talk about coffee, we're talking about a genus Coffea with about 120 species in it. And of course, the three main ones that we use for drinking coffee or commercial coffee are Arabica, that originated in Ethiopia, in the highlands of Ethiopia, and that is our most valued coffee product. Coffee Canephora is often referred to as Robusta. That originated somewhere in East Africa, probably around Uganda or the DAR, but possibly extending as far as into Angola, some reports count. And then we have Liberica coffee, which probably originated around modern-day Liberia, may have extended a little further into the center of Africa. Liberica coffee is a very, very small percentage, probably about 2% of global output. Most global coffees are Arabicas and Robustas. Now, our limitations to coffee production, number one limitation until very recently was always disease. We now have climate change, of course, playing a factor in limiting crop production. And these two are somewhat inextricable, as I'll show you a little bit later. But as far as diseases are concerned, the number one limiting factor to coffee production is this disease that we call coffee leaf rust. And so that's what I'll talk about today. It has been a problem for about 150 years and continues to cause devastating epidemics to this day. So a little bit of history. Coffee, as I said, Arabica coffees originated in Ethiopia and this region called Kaffa. And there are definitely anecdotal evidence that coffee, or at least an infusion of beans and sometimes leaves, was drunk in that area of the world for a long time. We also have lots of evidence from early exploratory studies that many of the tribes in Central Africa and into a little bit of East Africa chewed what we now know as Robusta beans as well. So we have lots of early uses of the crop, but these were not cultivated uses. These were uses where they grew natively. The first cultivation we see of coffee is in Yemen. And the Yemen brought the crop over, of course, from Ethiopia. The port of Mocha became synonymous with coffee at one point in time. And from there, the Yemen peoples traded coffee all throughout the Arab world. They retained a very, very tight restriction on the product. They would not allow beans or seedlings or anything out of Yemen, so all the production occurred there. But through the Arab trade, we have coffee becoming very popular in places like Turkey and Egypt. And eventually, through contacts with the Byzantine Empire, coffee is brought to places like Vienna and Venice and becomes very popular in Europe. Now, Europe, as coffee's rising in popularity in Europe, the Europeans saw a necessity to try to gain control of the growth of, or the cultivation of coffee themselves. And we see the first of numerous agricultural smuggling activities occurring at this time. The very first smuggling event is of a pilgrim from India who smuggled 6 beans out of Yemen in his turban into India, and began the first cropping of coffee outside of Yemen, which occurred in India. Very shortly thereafter, some Dutch managed to smuggle some beans up into the Netherlands, grow them in the Royal Botanical Gardens there, and then use that stock to start populating in their various colonies and cultivating coffee. So already we've got a genetic bottleneck here, because most of the coffee in India is from just 7 beans, and that in the Netherlands from just a few cuttings. But we've got the Dutch transporting then coffee to most of their colonies, and especially a heavy center of coffee production is the island of Java, which of course also becomes synonymous with coffee itself. The Dutch also bring it to Ceylon and other parts of Indonesia. And India, of course, is growing it as well. Now, at this point in time, the Dutch control all the production of coffee, and Europe is still clamoring for more coffee. So we have a couple of more smuggling events. For reasons known only to himself, the king in the Netherlands gifted a fertile coffee plant to the king of France, who grew it in his botanical gardens, and a piece of that plant was smuggled to Martinique. The story is that the merchant who smuggled it was running low on water rations and would use that to feed his plant rather than himself to water his plant, so that he could make it all the way to Martinique. But almost all of the coffee grown in the Americas originates from that one cutting that originated from a single plant in France, that originated from just a few beans in the Netherlands. So we have very limited stock there. After that, it's a heyday of smuggling. We've got a Portuguese diplomat who goes to French Guiana, sleeps with the wife of a diplomat in order to get his hands on some cuttings and bring them back to Portugal, etc. And pretty soon after that, for the next 100 years, we have coffee grown by all the major colonial powers throughout their colonial holdings there. Europe, of course, has experienced 100 years of heavy caffeination. Now, things are looking really rosy. By the mid-1800s, England, of course, has taken control of India and Ceylon, and they have turned every single square acreage available in Ceylon over to the production of coffee. So they are producing just an entire island full of coffee. And then disaster strikes. In 1867, a weird disease is reported on coffee in Ceylon that is devastating the crop there. And just within a few months of that report, we have it reported in India, where it's also devastating crops. That disease sample, and this is a picture I took at Kew Gardens of that original sample that was sent from the island of Ceylon to Kew Gardens in the UK for identification. It was identified by a very famous mycological team called Berkeley and Broom. But Berkeley recognized it as a rust fungus at the time, but as belonging to a genus and species that had not been previously known to science. And he gave it the name, excuse me, Hemileia vastatrix. Hemileia means half smooth, and that's the description of the spores. The spores are half smooth. And vastatrix means destroyer. So it's the half moon destroyer. Within a few years after that, the rust is reported in Indonesia and completely devastates the Dutch. And then by 1879, the British had to give up all coffee holdings in Ceylon. The plantations were abandoned, the plants were ripped up, and they turned to planting tea, basically, on the island. Ceylon then becomes a big producer of tea. The Dutch responded a little differently. They experimented with other coffees like Liberica, and in fact, most of the Liberica produced in the world today is still produced in Dutch Indonesia because that particular species showed more resistance. But by this time, the epidemic is well on its way. And what we're looking at now, this is an early map in 1952, showing the disease, the coffee disease part of the world and the undiseased. And within 2 years of this map being published, Liberia and the Ivory Coast both succumbed to the rust. So everything in the so-called old world was heavily diseased, but the new world remained disease-free, and it was believed that the Atlantic Ocean would form a big enough barrier, a cordon, to prevent incursion of the rust into the Americas. Of course, that didn't happen. And in 1970, rust was first spotted in Brazil, and then very rapidly moved its way up to the rest of the Americas. And by 1983, basically every region of the world where we grow coffee was infested with this rust fungus, except for a couple of farms in Australia. Australia's managed to keep it out, but there's only about 50 farms there through quarantine. And Hawaii, which this is not to scale. Hawaii is actually, of course, much further off the map there. The way the world responded to coffee rust, as I mentioned, in Indonesia and the Dutch colonies, they'd experimented with other types of coffee. In Eastern Africa and other parts of Africa, they turned to Robusta instead. But what's interesting about this picture, Brazil also turned to Robusta, but the rest of this part of the Americas continued to grow Arabica. What we saw in the early stages of the disease, this mass devastation where plants were destroyed, we didn't see so much in the Americas. It was a manageable level of disease that could be kept under control. We're still not entirely certain why the disease should behave differently in the Americas, but again, it was manageable. And so Arabicas, most of our good Arabicas are still coming from the Americas up to this, even through this point in time. The rust continued to move. In 2020, it hit Hawaii. And then very recently, we also found it on a couple of backyard coffee plants in Florida. So these are not commercial plants. These are plants by backyard ornamental plants, but it's still there in Florida. So again, it is everywhere except for a couple of farms in Australia. What really changed and drew me into this problem was what happened in 2012. And remember I said that in the Americas, we had not seen the devastation that we saw the rest of the world when the rust was introduced. But in 2012, that changed. Even though the disease had been there for a couple of decades, suddenly we had massive epidemics and complete crop loss. Up to 100% crop losses in some of these places in Central America, like Guatemala and El Salvador, and as far away as Jamaica and Colombia and southern Mexico were all part of this massive epidemic. This was our first really severe epidemic of the disease in more than 100 years. And again, the devastation was really outstanding for some of these farmers in Central America during this period. So the impacts of the disease, we've seen that cultivation techniques changed for parts of the world. Ceylon, for example, the British stopped growing coffee and converted entirely to tea. The more modern epidemic, the big rust, it's still impossible to calculate the overall damage that was done during this epidemic. But we do know that food insecurity was a really, really big problem in this part of the world during this period because, again, there was complete crop loss in some places. And there are estimates that a lot of emigration to northwards to other countries, including the US, was a response to this loss in crops during this period of the big rust. All right, so that's your little bit of history background to the disease. Now I'm gonna switch gears a little bit and talk about the rust itself. So this is a sorus in the background that you're looking at called a uridinium. I'll explain that in a second, and the spores of the rust on it. But as we mentioned a little bit earlier, one of the world centers actually of rust study has been Purdue University since 1888 with a man named Charles Arthur. And it continues to be a global center for the study of these fungi, which can be quite beautiful. These are all plant pathogens. These are different rusts here, but cause some of the most notorious agricultural and environmental diseases that we know of. Most of our plant disease is caused by members of the kingdom fungi. About 85% of our plant disease are caused by fungi. And this single order that's called Pathogens, that's the order that contains the rust fungi, alone contains about 8,000 species. That's a lot of species. Primates, the order primate, contains about 500 species, just to put it a little bit in perspective. It is the second largest order of fungi, and all of the members of this order, again, the order's name is Pathogens. We call these rust fungi, but all the members cause disease in plants. These are vascular plants. Most vascular plant families have members that are susceptible to disease by rust fungi. The group has a very broad range, but of course, individual species are very host specific. So they're highly specialized for whatever host it is or host that they infect and have narrow host ranges. And then there's some special terminology that we use in the study of rust fungi. A sorus is the particular pustule shown here that you can actually see. So this is a cross section through a plant leaf, and this part sticking out, that's the sorus. It's producing spores, so we can actually see that on the surface. But within the plant leaf, there's still a lot of these cells. These are called hyphae cells that are penetrating into the host cells. And all together, that fungal individual, the body of it, we call a thallus, just because there's special terminology in mycology since they don't form typical bodies like we're used to in plants and fungi. So that's the thallus and the sorus producing spores. Now, the rust fungi are really, really fascinating. They have many characters we don't see elsewhere in fungi, characters that we see rarely in biology. So they're a really, really good study system if you're interested in evolution of characters. They are what we call obligate biotrophs. That means they must have a living host to get their nutrition. This is very different from most of our other fungi, which can be facultatively pathogenic. These have to have a host. This limits the ability that we have in studying them, however, because we can't study them in the laboratory and culture or things like that like we do for other fungal organisms. They have the largest genomes that we know of in fungi. The genomes here, we're talking on the scale of gigs, 1.4, 1.5 gigs. Primate genomes are between 2 and 3 gigs, so it's not that much smaller than a primate. And again, to put it in perspective, something like Saccharomyces cerevisiae, that's the yeast, the brewer's yeast, It's a model organism. Its genome is about 20 megabases, so a complete order of magnitude smaller. So these are really, really big genomes. Again, limits our ability to do genomic work. They produce an alternation of generations. You guys know what that is? So that's when you actually produce different parts, different functions of life cycle in different thalli at different times during the life cycle. So instead of producing one thallus, they produce two separate thalli, one individual, to complete the life cycle. And what you're looking at on the right side of the screen at the top is what we call a gametothallus. That's one stage of an individual that's on an apple. And under that is the same individual, but a different stage of its life cycle on a different host. Here it's on a juniperus, and that's the other thallus it's forming. So this kind of complex life cycle is seen almost never in fungi. It's seen in a lot of animal pathogens. In parasitology, you'll find this kind of life cycle. But in plant pathology, this is the only thing that produces a complex life cycle with host alternation. This life cycle has 5 different stages to it. And because of this complexity, the hugeness of the group, difficulties in studying them, we actually don't have complete biological data for the majority of species, despite the fact that they're so important to agriculture. That's what a life cycle looks like, simplified. I won't walk you through this whole thing. But basically in uridanology, we use this shorthand to denote different parts of the life cycle. So a Roman numeral 3 just means the teleal stage. You can see here the sporothallus on one host and then alternating to a gametothallus on the other host. The really crucial parts of this life cycle are these. The spermagonia is the place where fertilization takes place. So this is where you have sexual recombination. And of course, that's very important for providing new genetic potential. And when pathogens are often able to overcome host resistance, it's because they're able to stack what we call these virulence genes in new combinations through sexual recombination. The other important part of the life cycle is this one here, and this is called the uridinyl stage. This is the only spore stage that's able to reinfect the same host species it came from. And so this is the stage that we usually find associated with epidemics, this uridinyl stage. And in this uridinyl stage, where it cycles on the same host over and over, a single spore can turn into billions of spores throughout a growing season. So you can see how this is really, really crucial to build up of inoculum and epidemics. Now, in coffee leaf rust, the only stage we know is the sporothallus. We don't know the gametothallus stage. And the only stage that's really well studied is that uridinyspore stage. Again, that's the stage that causes the epidemics, where those uridinyspores germinate, reinfect the same host, produce many more sori that produce spores and regerminate. And that cycle can continue under the appropriate environmental conditions almost indefinitely, as far as we know. Now, with this rust, we do know that occasionally it will produce other parts of the sporothallus. So occasionally it'll produce telea. Those telea germinate and produce basidiospores. But we have no idea what the function of those basidiospores are. They don't reinfect coffee. What hosts they actually jump onto to infect is not known. But of course, it's this unknown host in which sexual recombination via fertilization would take place. So it becomes very important for understanding epidemiologically what's going on with this particular organism. And so the research questions we have, or at least that we'll look at today, are what is the reproductive biology? Does Hemileia vastatrix undergo a sexual stage? Because that's going to lead to different breeding strategies than something that's just strictly reproducing clonally. Where did the pathogen originate? We're going to look at some epidemiology. How does it spread? And especially what happened to cause that big rust epidemic, and how can we use that hopefully in predicting or avoiding similar epidemics in the future. So as far as reproductive biology, there are two competing hypotheses. One, that the rust is sexually reproducing and we've just never observed the sexual state. And the other is that it's become specialized and reproduces strictly as a clonal organism. And again, knowing which of these is the correct answer does have implications for how we try to control it and breed coffee against it. And one of the best ways to test this type of reproductive question when you can't force reproduction in the laboratory is to use a population genetics approach, which is what we did here. So we used, back at the time we began this study, something called microsatellites were the most sensitive markers that we could use to try to determine individual level genotypes and get at whether this was clonal or sexual reproduction. So to develop microsatellites, you get a genome, and then you search that genome for sections of what we call little mini repeats. I'm showing an AT here. You develop primers for that. You screen a small subset of your population, see if your primers are variable, and then if they work well, you can screen a large population and get basically genotypes similar to a fingerprint for each individual, and then do analyses on that. This is Terry, my postdoc Terry, in the field in Panama, and what we're doing there, this is an infected coffee plant, and we've rigged up a really heavy vacuum at the end of this little thin needle here, and we use that vacuum in the field just to suck spores from a single sorus out into a tube, and that way we can genotype individual sori in the field. It's really a pain to get that thing through customs because the battery's like this big, so everybody always thinks we're trying to carry a bomb in our suitcase, but at any rate, this method has worked really, really well to get these samples for analysis. And our first sampling consisted of 105 isolates from these 12 countries that I've highlighted here. And these were all isolates that we collected in the field between 2014 and 2016. So this was immediately after the worst of the big rust when we started collecting these samples. And doing all these different types of analyses on the samples, basically, and I'll just only walk you through a few, but the one on the top is something called a linkage disequilibrium. And if your hypothesis is one of sexual reproduction, of outcrossing, then you would have an expected allelic distribution that would cross these blue lines. And you can see that in all these cases here, and I'm just showing a subset we analyzed by country, by host, by world region, and globally, that none of these populations that we analyzed have any hallmarks of sexual reproduction by that measure. We did statistical testing, also a network analysis here on the right, all different ways of asking the same question or testing the same hypothesis. Is there any markers for sexual outcrossing? And the answer is no. At least within the sampling that we have, which is somewhat limited, there is no sign of any sexual outcrossing. This network analysis is particularly indicative. If you had sexual outcrossing, you would see a whole lot of reticulation in that tree, but we see strictly, for the most part, clonal reproduction. What we do see in this network analysis is two very large, what we call MLGs. That's just a multi-locus genotype, but these two genotypes make up the majority of our samples. In other words, most of the specimens we collected around the world belong to only one or two genotypes. So again, very, very little variation here in that data set. One of the genotypes on the top there is mostly of African origin with some American, and the one on the bottom is almost exclusively American in origin. So what is the origin of the rust? where did this epidemic first get started? Or the rust, I should say, itself first come from. The hypotheses in the literature, there are two. One suggests Ethiopia. Ethiopia, of course, is the home of Coffea arabica, and it makes sense that people would hypothesize that would also be the origin of the disease, and then it was then moved with coffee out of Ethiopia. And then there's alternative hypotheses that Sri Lanka was the center of origin, and that when we moved coffee to Sri Lanka, it jumped onto the coffee there. And that's what began the epidemic. So two very different ideas about how this epidemic might have been started. But basically, we can use the same sort of approach, a population genetics approach, to try to test these hypotheses. But you can see, first off, that there's some serious holes in our sampling. For instance, we didn't have Sri Lanka. We didn't have Ethiopia, and we were missing some other obvious players like Brazil and parts of Indonesia, where the rest moved on next. And so to get some of these holes filled, we actually turned to herbarium specimens. The Arthur Herbarium has a huge collection, not huge, but a significant collection of coffee rust. I was also able to do sampling at Kew in the UK and at the National Fungus Collections in Beltsville to get some historical specimens so that we could test these origin theories. In the end, to date, we've got about 826 isolates. We've expanded our sampling to include the regions that we want to test for origin, and gone back at least to 1906 to look. So we still haven't cracked the 1800s here, but we've gotten as far back at 1906 to look at what some of these early genotypes look at. So first off, here's the network. So that's that same network analysis, just enriched now. We see the same pattern, 2 MLGs, so 2 really dominant genotypes throughout most of the world. One primarily African and one almost exclusively American. And first, let's look at the hypothesis that the rust originated in Sri Lanka. All of our Sri Lankan genotypes belong over here, some to MLG28 and some as variants of MLG28. But there's not a lot of variation there, and we don't see any ties to where we would expect them to come from Africa or elsewhere. When we look at Ethiopia, we see even less diversity than we saw in our Sri Lankan isolates, which was a real surprise. But the Ethiopian isolates almost all belong to single MLG20 with a few variants on that, and no connection really to Sri Lanka from there. So if the rest originated there, it didn't make it to Sri Lanka, and if it originated in Sri Lanka, there's no direct link back to Africa for it. What's interesting in this network is this little tail that you're looking at here. Every one of these dots represents a different genotype. So we have a huge number of new genotypes here, and they all come from a single area on this map. They're all coming there from around Lake Victoria. They're coming from Kenya and Rwanda. And so there is a school of thought where the way you find a center of origin for an organism is to look for the center of diversity. So another way to sort of present these data is to just look where the variation in this total global population is. So here, the size of these white circles represents the amount of variation, and inside are the numbers of samples tested. So for instance, in this Lake Victoria region in Rwanda and Kenya, we had 23 isolates at the time we did this sampling, and every one of them represented a different genotype. The next area of diversity we see is actually going 2 ways, one over to West Africa and one out here to India, Sri Lanka, and Indonesia, and then very little of the global diversity residing in the Americas. And so that sort of points us at actually the Lake Victoria region as the center of origin for this rust, not Ethiopia, not Sri Lanka. Now, if you recall, when I was talking about where hosts were, Arabica, of course, doesn't grow naturally around the Lake Victoria region. Other species of Coffea, however, do. So another big hint, at least to me, is biologically. I have collected this rust from India to Jamaica to Hawaii, and I have never seen it produce telea in the field. Telea is another part of the sporothallus that is produced right before usually for a rust, at the end of its life cycle or its seasonal cycle before it produces basidiospores. But I saw a lot of natural telea production in Kenya about a month ago. And that hints to me that the conditions there are very conducive for this thing to complete its life cycle in that particular region of the world. There's another hint about this just in the symptomology of the disease. When you see this type of rapid onset, sudden epidemic that's really, really devastating, that's usually indicative of a host jump. And that's that kind of severe disease progression, when a pathogen that's co-evolved with one host suddenly jumps to another one that is what we call naive, and then causes very, very severe. So to me, the symptomology always hinted that it didn't originate on Arabica, maybe another host, and then jumped onto Arabica in Sri Lanka. So the question then is, what species of Coffea might it have originated on around Lake Victoria? And so here's our map again of these three species that we use for cultivation: Arabica, Canephora, and Liberica. And of course, Canephora is native to this part of the world, at least to the western side of Lake Victoria. There's one more species that is native to this part of the world as well. This is Coffea eugenoides. We don't really use this commercially, but it does grow here, and this is a healthy plant. While I was in Kenya a month ago, this is another eugenoides that I found that is completely defoliated. So doesn't seem like that would be a good host, because again, it's behaving when it gets contact with the rust in this completely devastating manner. So what about Canephora? Well, Canephora actually fits quite nicely with what we know about the biology of the pathogen. It originated first off in the tropical lowlands, whereas eugenoides and Arabica originated in the highlands. If you know anything about coffee production, you know that when you move up in elevation, disease pressure is much less. That's because the pathogen does not like higher elevations to anthropomorphize. It prefers lower elevations. So it seems that it would have evolved probably in a more lowland habitat. And the reason Canephora, even though it tastes horrible, the reason people cultivate it is because it shows partial resistance. It doesn't become defoliated or completely disease-ridden. Yes, it will get disease, but it's like a common cold to Canephora as opposed to completely devastating disease. So Canephora seems like our most likely suspect at this point for origin of the rust, and then at some point it was came into contact with Arabica, jumped onto it, and caused devastation. So is there any evidence at all that we had rust in that region of the world before it was officially recognized? And if you start reading a lot of these early historical accounts or even old histories about coffee rust and coffee, you will often find this reference over and over again, worded almost exactly the same. In 1861, a British explorer found what later became known as coffee rust on a wild species of coffee near Lake Victoria. I have traced every mention that I can find in the literature back to dead ends. They're either self-referential or they just make a statement without a reference. I've never found the original source. But I started digging around again. Actually, when I was in Kenya, I started reading about early explorers around the Nile. Around Lake Victoria, because remember, this is when people were trying to find the source of the Nile. And these guys fit the bill really well. They're British explorers. They're around Lake Victoria in 1861. They're actually the first people to discover the source of the Nile as northern Lake Victoria. And it turns out that James Grant was a botanist, and he published a lot of his botanical findings from this region. And I called a friend of mine at Kew, and he found me a specimen of Grant's coffee there in Uganda that he collected in Uganda and made the notation that this was being chewed. Right now, we are looking through all of his ephemera to see if we can find out where that statement that he saw rust on this plant came from and definitively pinpoint it. But it does look like rust was at least likely to have been present there on Robusta. This is definitely Robusta that Grant collected during the right time frame and before it became an epidemic on Arabica. Oh, that's Aaron there. So how did the disease spread? The two prevailing hypotheses here are that it spreads via trade winds or via humans. So here is basically a reconstruction of what our data are showing us for the spread of MLG28. We have it moving out of Lake Victoria region into India and Sri Lanka, and then on around the world from there. And this is the same MLG that made its way to the Americas. And then here's the other MLG, what I call the African one, but it went the other direction. And this one is different, but it moved back into West Africa. And then surprisingly, we see traces of this MLG also being introduced into Brazil and, oddly enough, Puerto Rico. And that Puerto Rico one's a little bit of a head scratcher because, at least within our limited sampling, the sample we have of the African genotype is older than we have of the actual MLG28, which is supposedly moved that way into Puerto Rico. But our map does more or less map what we know disease progression was, with the exceptions of those two anomalies there. So let's look at how this could have spread. We know that there was something called the Indian Ocean Trading Network, which was long established in this region of the world. And at the time of the first outbreak, Kenya, Ceylon, and India, the British were using this Indian Ocean Trading Network very, very heavily to trade between their three colonies there. And there are all sorts of examples of people using leaves as packing materials. They could have been shipping around infected seedlings. It doesn't even have to be that complicated. Remember those spores? They're really spiny and they cling to your clothing. That's a shoe after walking through a rusty field, just covered with spores. So it's very easy to pick these things up inadvertently and carry them around. But the simultaneous display of disease in these two parts of the world, again, being connected very, very closely through the British trade routes to Kenya, also would seem to suggest that it was probably a human event that caused that initial outbreak. The one that we can say for certain is the introduction into Hawaii. And we examined trade wind routes as well as doing population genetic analyses there. And definitively, I can say, or at least 99% certainty, that the isolate that started the epidemic in Hawaii came from Central America. It did not come through trade winds, which were blowing in the other direction at that time of year. Hawaii has a long tradition of using migrant workers from Central America during coffee picking time, so it would have been quite easy to just transport spores on an infected piece of clothing. This Puerto Rican mystery, looking through a lot of our old archives in the Arthur, we came across this letter that was written in 1920s. It was written to Charles Arthur, who must have asked a question to someone about a possible introduction into Puerto Rico. And this is from an actual agricultural station specialist in Puerto Rico, who notes that he did receive an infected bunch of coffee plants in 1906 that had coffee rust, and he hoped that he had eradicated it before it spread throughout the island. So uncertain if that's the origin of this mysterious introduction, but certainly these types of things could have happened. So the big rust, what happened? The biggest hypothesis is that we had the evolution of a new race. In plant pathology, we have something called the disease triangle. And this states very, very simply that in order to have a disease outcome, you need a competent pathogen, you need a susceptible host, and you need a conducive environment. You have to have those three things before you have a disease outcome. We know that the host did not change during the big rust. These farmers were using the same cultivars they had been using. And we see no evidence for any different genotype. The proportion of MLG28 may have changed in response to selection pressures, but its presence had always been there. That leaves a conducive environment. So did the environment change to cause this epidemic? And these are maps downloaded from NOAA. It's hard to get these types of data with granularity. But basically, pre-epidemic, we see pretty much normal temperatures in this part of the world. And during the epidemic, we see elevated temperatures. And then with rainfall, pre-epidemic normal. During the heavy epidemic year, we have increase of rainfall. So what does increase of temperature and increase of rainfall have to do with creation of an epidemic? And basically, it goes back to the biology of this particular rust. Every fungus is different, every rust is different. This particular rust, one spore under a normal growing season of 3 months can produce about 300,000 spores. So there's an epidemic potential there if it's not controlled. But usually the environment controls this to some extent. This germination stage requires 100% water potential, rainfall, high humidity, in order for the spores to germinate. Otherwise, no infection gets established. And then the colonization rate, the rate it takes for those hyphae to ramify through the host and then produce a sorus, is dependent on temperature. It has an optimal range of 20 to 28, but it turns out the warmer the temperature, the shorter the generation time in order to ramify. So what does that mean? Basically, the difference between a 20-day cycle and a 16 cycle means the difference in the same growing period, give or take, between producing 300,000 spores and about 90 billion spores. So just a few changes in temperature. So that's it. We have lots of future work planned, and I do want to thank actually all of the coffee farmers around the world who have helped us with this. Thank you very much for your attention.
Cathie, I know President Chiang had to take a phone call, so we do have time for a few questions. So if you've got questions, please ask Dr. Aime some questions.
So you said the genetic population of coffee in Brazil and South America originated from one very narrow
One cutting.
cutting. Has that changed? Have more genotypes been introduced?
Yeah. So it has changed in modern times. There's more grafting, trying to graft with Robusta stock. There are more cultivars, although most of those are being developed in the Americas for the Americas. They're still not importing a lot of cultivars from outside the America. But there is a little improvement to the stock. But even the stock from other parts of the world is very limited as well.
I'm wondering, what is the role, in your opinion, for remote sensing to help with rapid assessment of disease
I think remote sensing could do a lot because it's all about buildup of inoculum. It's about keeping it from doing one more generation. If you can see on the ground that you've already starting to get a buildup, you could, if you had the resources, go in and spray, for instance, to cut that inoculum back or do some type of sanitation.
And to add to that question, how long it takes first the disease symptoms appear versus devastating the entire?
Yeah, so a great question. It starts out slow, so under the right temperature and humidity regime, you might see a few spots and then 2 weeks later have a full-blown epidemic, again, given the right environmental conditions. So it could happen very, very quickly. Sometimes you can just, in a self-managed farm, use sanitation to cut some of that inoculation down. That cuts that growth rate down as well. But uncontrolled, yeah, 2 weeks can be a difference.
I have a question about the scale of the great event that you have.
The big
event.
rust, yeah.
And then you mentioned the Hawaii data set where there was an infection. Now, is there some simulation technology that would allow you to look at the patterns and say, like, if there would have been more land mass and more production in the Hawaii area, it would be like similar event? So that's one part of the question. The other one is, how vulnerable is Australia?
Yeah, well, Australia has, if you've traveled there, they have really tight quarantine. But, you know, Australia and New Zealand had tight quarantine against Puccinia cityi, which is something that kills eucalyptus, among other things. And it still did manage to get there. So right now, they have strong quarantine, which seems to be keeping it out. The production in Australia is really limited. There's about 50 farms involved. So even if someone were to inadvertently bring it in, they would still have to carry it to one of these small farms. But yeah, how good is their quarantine is what that one boils down to. Now, your first question about the big rust and would we have seen epidemics in Hawaii, like we saw earlier. So one of the things we're working on right now, and it's taken actually a couple of years to set this up, is to be able to do really controlled experiments on leaf discs to look at and pinpoint and quantify the effect of different environmental factors on spore production, the amount, source production, and generation time. And we've almost got the last piece of equipment we need to full scale ramp that up. But then we will be quantifying exactly what a change in degree means in change in generation time. Then you can use that to model with real world data, go back and look, for instance, at Hawaii, see what the exact growth conditions were then, if they were less conducive to an outbreak or an epidemic just because the conditions were not quite warm enough.
And I have another follow-up question, sorry about that, it's directly related to that. To what extent could you, if you have a controlled agricultural environment, protect actually the plants and the disease from spreading? To what extent do you have to go to another outbreak?
To protect a plant,
Yeah.
a specific individual plant or a plant species?
A production, let's put it.
A production. The best, you have to go to pretty heroic extent in most cases. So for individual high value trees, plants, you can inject them with fungicide. You have to do this periodically, but that's a method for doing that. It's not practical in a situation like this. We mostly for a farmer to absolutely save his crop, you're talking about spraying copper fungicides. It takes him off the organic market. It requires a lot of expensive equipment and manpower and the ability to obtain the fungicides to get them sprayed. So it's much less practical in most of these situations. Now, Brazil and Vietnam have managed to mechanize Robusta, and they've got a really good routine of prophylactic spraying, et cetera, to protect their investment in those crops. But that really needs an ability to mechanize production.
Thank you for your presentation. It was beautiful and terrifying for a coffee. But I think what struck me was across the stories you were telling, you were following the footprints of both humans and nature, identifying how to share plants, information, and disease. And one of the things that came out for me, at least, was this idea of we don't have a complete biological picture of the rust. And so this massive data collection initiative in remote places at scale. Do you think there's a place for things like citizen science or alternative modes of public participation to help scale your research?
Yeah, absolutely. And we do use, not for this project, because most of our strongest citizen scientists are usually in European countries where we don't grow this particular crop. But we have used them extensively for some other, we did the first continental fungal foray, for instance, a couple years ago, all depended on citizen scientists, training them, providing them materials, showing them how to collect and record data. It's a really good idea. Yeah, it's a good idea if I could figure out a way to mobilize them into other parts of the world. And here you're talking about also interaction with coffee boards, et cetera. It gets a little more politically complicated, each of these countries, but it's a good idea. Thank you.
We're at 5:30, so Professor Chang had to take the next phone call. So I think let's thank Dr. Aime and Catherine. Do we have time to have a few more refreshments before people go and continue some conversation? So let's thank you one more time.
Thank you.